All right, good evening, everyone, and welcome to Genomics Lite. This is our CRISPR in Focus session with our guest speaker, Olivia Edwards, who is a PhD student at the Sanger Institute. My name's Em. I'm the Education and Welcome Officer, Education and Learning Officer at the Wellcome Genome Campus. And I'll be hosting this evening session, keeping us all on track and sorting any tech issues if we have them. So before we kick off, I'll do a little bit of housekeeping. So all the events that we host aim to help everyone explore what the science of genomics means for our lives. So we invite you to ask questions this evening, both about the science and about Olivia's career journey, but we ask that you do so respectfully. And I will be moderating the platform this evening to make sure it's safe and inclusive for everybody. In case you've not used Zoom webinar before, there's a few different ways that you can interact with us. You can't use your camera or your microphone, but you can leave us a message in the chat box, or you can ask Olivia a question in the Q&A box as well. We have closed captions available on this call in case you want to use them. If you do want to use them, you can press the CC button at the bottom of the screen, and you can move them around as well. So if they're in the way of something Olivia is showing you, you can move them around the screen. Likewise, if you don't want them, you can get rid of them as well, but if you'd like them, they are there. We'll also be using polls during this webinar, so they'll pop on your screen and ask you a question. If you're having any issues seeing them, you can just leave us a message in the chat box instead. So we are streaming this webinar, theoretically at least, from the Wellcome Genome Campus. We're a large scientific research campus based just south of Cambridge in England, and we're made up of a few different organisations, the main two being the Wellcome Sanger Institute and EMBL EBI. The Wellcome Sanger Institute is where Olivia works and does a mixture of what we call wet lab as well as data science, whereas EMBL EBI, the EBI bit standing for European Bioinformatics Institute, centres around data science, so has no wet labs, is all computer based. It's lovely to know where people in the audience are from, so if you would like to let us know, you can pop a message in the chat box. It's fun to see how far away from Cambridge we've got people joining us from. Obviously, all the work that we do on campus is around genomics, that's in our name. And so it can be useful, obviously, to start by understanding what we mean by genomics. So we think about DNA, we think about genomes. A genome is the complete set of genetic instructions of an organism. And on campus, we use this to understand different things. We use it to understand the relationship between different humans, between humans and different species, but we can also use it to understand the relationships between different cells. And as Olivia is going to talk to us about tonight, we can also use it to understand how diseases work and how we can manipulate DNA to understand disease better. So the rough sort of running order of this evening's talk, Olivia is going to talk to us for about half an hour about CRISPR, and we'll then have time for a Q&A with her about the topic of CRISPR. She'll then talk to us a little bit about her career journey. So she's currently a PhD student and is going to tell us a little bit about how she got there. And again, we'll have a bit of time for a Q&A on that, and we'll finish by 5:45 this evening. If you have to leave before then, this session's being recorded and will be available on our YouTube and on our website either tomorrow or early next week. Before I hand over, Olivia also very kindly wrote for us a new article on our Your Genome website, this one here, all about how we can use CRISPR to study diseases further. So this will be part of your learning pack that you'll get sent either tomorrow or on Monday from Eventbrite, but it's a really great article that is a great summary of what CRISPR is and how we can use it to study disease. So that is all from me, so I will stop sharing. I will pass over to Olivia to tell us more about CRISPR.
Great, thank you for that introduction, Em. Is that just double check that I'm showing the right screen?
Yeah, that looks great.
Great, okay. So yeah, as Emma's introduced myself, I'm Olivia, and so today I'm going to be talking about the gene editing technique known as CRISPR, which recently has become a hugely popular topic of discussion in the scientific community and sort of in wider society as well. So I'm going to focus on how we can use this technique in the lab to specifically tackle diseases, and how it might also be applied to other important issues as well. So to start with, I'll sort of outline what is meant by gene editing. discussing the structure of DNA, how it changes, and why these changes are important for us to investigate and understand. Next, we'll look specifically how CRISPR works and how it works in the lab. And finally, how CRISPR can be used by scientists for research and beyond as well. So after a Q&A session, I'll then talk about my journey through education as well, and how I chose to study a PhD at the Wellcome Sanger Institute. And sort of, if anyone's got any questions more about that, they can ask in a Q&A after that part as well. So let's start with an introduction to gene editing. So firstly, we've set up a poll just to see how many of you have actually heard of CRISPR before. So some of you might have discussed it in your classrooms perhaps, or you might have seen it on the news, or maybe this is the first time that you've heard of it. So yeah, if you'd like to answer in the poll, then that'd be really interesting.
So far, most people have heard of it but don't know much about it. We've got a couple people that have know quite a lot about it, and a couple people that have never heard of it. So a nice mixture, but most have at least heard of it.
Okay, great. Okay, so to start this topic, we first need to sort of understand what DNA is and how it's structured. So within most eukaryotic cells, including most cells found within the human body, DNA is found in a structure called the nucleus, where it's tightly wound and packaged into structures that we call chromosomes. And DNA encodes all the genetic information of an organism, and so it forms the blueprint of making all the proteins required to build and maintain these cells. And so it's a very important part of any organism. So it's important that we are able to understand it. So the instructions for making these proteins are encoded in different sections of the DNA that we call genes. And each gene has a different DNA sequence, which instructs the cell to produce proteins with different structures and different functions. So up close, we can see that DNA forms a structure called a double-stranded helix, which with each of these strands made up of a sequence of nucleotides. And there are 4 different types of nucleotides: guanine, cytosine, adenine, and thymine. And these nucleotides form specific pairs which allow these 2 strands of DNA to join up and form the helical structure. So for every guanine on one strand, there's a cytosine on the other, and vice versa. And in the same way, for every thymine on one strand, there's an adenine on the other, and vice versa. So when building these proteins, these bases or nucleotides are read in 3s. So each combination of these 3 nucleotides encodes for a different amino acid, and these are the building blocks of proteins. So these amino acids are strung together in the order that's specified by the gene sequence, and it forms these chains which can then be folded into the proteins. So DNA is formed of long sequences of nucleotides, but these sequences aren't fixed and actually can sometimes change. And these changes are what are known as mutations. And there are many different types of mutations, some of which I've sort of outlined on this slide. So, for example, a substitution describes one nucleotide being changed in the other. So, such as this A to C mutation we see here. And alternatively, nucleotides can get added into the sequence or removed from the sequence in mutations known as indels or insertion deletions. So there are multiple different factors which can cause mutations to occur. And one way is during actually DNA replication, which occurs when a cell divides. So sometimes the replication machinery in the cell will make errors when it's copying it, and this will change the sequence. And although there are repair mechanisms in place which look to identify and reverse these errors, sometimes these fail and it leads to a mutation being passed on to the daughter cell. There's also multiple environmental factors as well which can mutate DNA, and these are collectively known as mutagens. So this includes UV light, chemicals found in cigarettes, and even some types of viruses as well. So we know that the sequence of DNA is very important. So for example, in protein coding genes, the sequence encodes the precise instructions of how to make the protein properly. So what happens if a gene actually becomes mutated? So in many cases, changing the sequence of a gene does not actually end up changing the final protein that is created from it. And this might be because the change in sequence doesn't change the order of amino acids that the protein is made from. Or it could be because the mutation occurs in a part of the gene that is known as a non-coding part. However, sometimes mutations will change the structure of the protein, and this difference might mean that the protein no longer functions properly, or even could allow it to perform a new function in the cell. Another effect might be on how much protein is produced as well. So there are sequences upstream of each gene which are required for it to be turned on and for its protein to be made. And so changes to this sequence might mean that the expression of the gene is either turned up or turned down. So what can these protein changes actually mean for an individual? So between individuals, we can see that there are many physical differences, and many of these unique characteristics, such as eye color, are driven by differences actually in our DNA sequence. And this genetic variation can also influence other characteristics which we don't directly see, but things such as disease susceptibility or drug response as well. So sometimes these differences are driven by just a single mutation, and other times these are the result of multiple mutations and even environmental factors interacting with each other. So for example, an individual's height, there are thought to be lots of different things that has an effect, some thought to be genetic and others non-genetic. So this brings us on to our first discussion question. And this question is, why might you want to research the effect of different mutations? So if any of you have any thoughts or suggestions, we'd be really interested to hear them, and feel free to add them in the chat box and we can discuss it a bit more.
So if anyone's got any ideas, pop them in the chat box and I will read them out for us.
Great.
So we've got to understand the function of specific genes, to see how to solve them. So I guess just to solve or to correct a mutation, I guess. Someone's put because they can lead to genetic diseases. So again, I guess to understand how the disease works, to understand how to treat patients. So I guess that links to the point you made about difference in drugs. We've got loads of great answers. So we've got lots around sort of, a lot about the sort of disease side of things. One that I really like saying, to see how they could be used to our advantage. I'm not sure what advantage we're thinking there, but I like the idea of that. To trace human migration and evolution. So we're thinking on a bigger level there. Loads of great ideas. Just having a scroll through. Someone's pointed out things like golden rice, so like deliberately making improvements. And someone's put to make artificial mutations that are beneficial. So yeah, loads of great ideas.
Great. Yeah, no, they all sound amazing and actually have pretty much ticked off all of the examples I have on the next slide. So that's really amazing that, already that you can come up with all these great ideas, and especially thinking on the lines of sort of using these mutations to our advantage and things like golden rice as well is sort of leads on to later on and sort of the potential uses of CRISPR and what CRISPR might be used for in the future as well. So really thinking ahead with these answers, which is great. Okay, so on this slide, I've just sort of outlined some areas of research which focus on understanding these specific effects of different mutations. So as we've said, we might want to use it to understand actually what a gene does and what a protein does. And so this can also help us understand how different biochemical pathways work in an organism. It also leads to a better understanding of disease mechanisms, as we said, as well as identifying individuals at risk of a disease and also ways of treating them as well. And I think someone else said also about evolution. So yeah, we can use this information from genetic variation to understand some really core, interesting parts of biology and understanding how we evolve. And for example, viruses evolved, and that obviously links on well to COVID and how we can trace COVID and things like that as well. So it's definitely like a very important subject that is sort of makes its way into almost every part of biology. So now we come on to how we study these mutations. So when it comes to studying them, there are two main approaches which both focus on the idea of comparing individuals with a mutation to those without it. So the first approach focuses on studying a population. So using DNA sequencing, we can look for individuals which carry a specific mutation and those who don't. And what we can then do is compare characteristics between these groups and see if there are any of these characteristics which correlate with the presence of a mutation. So this is a very powerful technique because we can directly assess clinical characteristics, for example, the prevalence of a specific disease. However, there are some drawbacks as well. So it can often be very difficult to find enough individuals with a specific mutation to be able to find correlations that are statistically significant. And it also doesn't inform us exactly what a mutation might be doing. So for example, how it is changing protein function, or if it is, and so this requires a deeper analysis in the lab to work this out on a molecular level. Across these individuals as well, there'll be a huge amount of genetic variation that isn't linked to the mutation of interest, so a lot of background mutations, and it can be quite difficult to work out if an effect we see is actually being driven by these background mutations instead. So what we can do instead is actually look at these mutations in cells that we culture in the lab, and this uses a method known as gene editing, where we can change a specific sequence of DNA to any mutation we might want to study, and we can look at what effect this mutation has on our cells. So for example, this might be a change in division rate, change in gene expression, or protein structure and function as well. And we can make sure that the only genetic differences between the cells we're comparing is the mutation of interest as well, which makes the statistical analysis much easier. We've got to remember though that using cells which are grown in these dishes in the lab might mean that the effect of a mutation behaves differently to how it does in an organism or in a human. So often geneticists try to integrate both types of studies in their research, and they're quite complementary approaches. So now we've talked about the concept of gene editing and why you might want to use it, I'll talk a bit more about the specific technique CRISPR and how that works. So the CRISPR-Cas9 system is a gene editing technology which allows us to alter DNA sequences through DNA repair mechanisms. Whilst there's been many other gene editing techniques used in the past, CRISPR is really popular in research today due to its high versatility, precision, and ease to implement as well. So the technology was actually adapted from bacterial immune systems, which have evolved in some bacterium to protect them from viral infections. So viruses which infect bacteria are known as bacteriophages, and these viruses inject their DNA into the bacterial cell and hijack the cell's machinery to produce and release more of these viruses. Some bacteria, however, have developed a way to prevent this infection. They've generated a system which specifically recognizes viral DNA and cuts it. And by cutting this viral DNA, the cell no longer produces these new bacteriophages, and so it stops the infection in its tracks. And in 2012, Jennifer Doudna and her team worked to repurpose this immune system to produce the CRISPR-Cas9 gene editing technique. And this work has been monumental in the field of genetics and was actually awarded the Nobel Prize in Chemistry last year, which is obviously a massive achievement. So to understand how this bacterial immune system was actually repurposed to a gene editing technique, it's important to think about what a gene editing system might actually need. So the first important component is something which is actually able to alter DNA sequences. And we can think of this as being the machinery of the gene editing system. There also needs to be something which guides this machinery to the correct sequence of DNA that we want to target. And finally, in some cases, we need a set of instructions as well, which tells the cell what the new sequence should be. So in the CRISPR-Cas9 toolkit, the Cas9 protein is this machinery, and it's able to cut double-stranded DNA sequences. And this is guided to the correct DNA sequence by an RNA molecule, which is appropriately named the guide RNA. And finally, a DNA template, which has the correct or the desired mutation, or what we can call edit, can also be added as well to act as instructions for the new sequence. So how do these components all come together? Initially, the guide RNA is designed so that its nucleotide sequence is complementary to the region we want to target. And by being complementary, it means it's able to base pair to the sequence of interest. And this opens up the region as shown here. So The guide RNA also has a region which is able to bind the Cas9 protein, and this means it guides this protein to the region of DNA. And Cas9 is an enzyme which acts as genetic scissors, and it's able to cut the DNA strands at this site. So now the DNA strand has been cut, the cell will try and repair it. It will recognize this break and it will say, okay, let's try and repair it. So it's at this point that we can instruct it to actually edit the sequence in different ways whilst it's repairing it. So the first type of repair involves a process known as non-homologous end joining, which in short basically involves sticking these two blunt ends of DNA back together. However, this process is often very error prone, and this means, for example, that random nucleotides might be inserted when the two ends are rejoined. It also means that alternatively and more commonly, regions of the DNA can actually be deleted before the two ends are joined back together. But this is actually sometimes very useful to us. So for example, sometimes you want to completely disrupt the function of a protein to see what effect this might have on a cell's behavior. And an easy way that we can do this is by creating a big deletion in the gene that encodes it. And so we might want to use this pathway for doing that. The second option, or the second repair pathway we can use, requires a template to be used as instructions. Sorry, I've gone too far. As instructions for the repair system. So the template will be a sequence of DNA which has regions matching the two ends, plus any desired mutations we want as well. And in a process called homology-directed repair, which occurs naturally in the cell, the cell will use this template to repair the DNA, adding in any of these changes to the sequence. And this is really
just had a quick question of what does homologous or homology mean?
Yes, of course. So homology is basically, if I go back a slide, it's used to mean that these sequences here are the same as these sequences here. So homology means like to match something. So the reason this is called homology-directed repair is because it's using this sequence which matches up to our cut DNA. And by being the same sequence, the cell's able to say, okay, this is what this should look like. So I'm going to use this homologous sequence to repair the break. Whereas the first option, the non-homologous end joining, we're not using this template strand. So we're literally saying we've got two bits of DNA that are broken. Let's not worry about whether they're meant to be together. Let's just stick them back together. So this is a sort of more messier mechanism, if that makes sense.
Awesome, thank you.
Great. So yeah, this can be really helpful. I can't remember if I said this. If we want to make more precise changes, so if just say we want to change just a single nucleotide in our sequence, we'll want to have a template which specifically has just this one change, rather than having the first method, which, you know, will either, it might make one change, but we're not really sure what it's doing. We can't control it so much. So we can see how CRISPR works when the components are actually inside the cell, but how do we actually get them there in the first place? So firstly, the CRISPR components need to be synthesized, and we can make the guide RNA and Cas9 enzyme outside the cell, and we can then couple these two together and put these complexes into the cell we want to edit. Alternatively, what we can do is actually generate DNA sequences which encode the CRISPR components, and we can put these sequences inside the cell. And then once inside the cells will actually transcribe the sequences and make the components themselves. But to actually get either the complexes or the DNA sequences inside the cell initially, there are multiple different methods that we can use, which I'll just run through because I think it gives quite a nice idea of actually visualizing how we do this really sort of complex mechanism in such a small system. So one method of getting the components inside is called electroporation. So this is where we use electrical pulses to create breaks in the cell membrane, and this allows the components to get in through where the cell membrane has broken. Another way is to actually package the components in a lipid capsule. So because the cell membrane is also lipid-based, these capsules are known as liposomes. They can actually fuse with the membrane, and once fused, the CRISPR components can then be released into the cell. And finally, another popular method is to do what's known as viral transduction. So here the components are packaged into virus particles, and these are used to infect the target cell where they can then be released. And then once inside the cell, the gene editing can take place as discussed. So now that we can do gene editing, what can we actually do with it? So as we sort of discussed earlier, the fundamental approaches to understanding the effect of a DNA edit is to compare a system with the edit versus those without it. And in the lab, this is something that we can quite easily do because we can compare our edited cells to the unedited cells that we begin with. So some things we might want to compare between these cells is, for example, their division rate, maybe differences in protein structure or differences in their gene expression and expression of other genes as well. And this can all be decided when we actually come to design our CRISPR experiment. So this leads on to our next discussion point. And this is, if we want to do a CRISPR experiment, what are the different things we need to decide on? So, imagine sort of putting yourself in the shoes of a CRISPR scientist, and you've got this really cool technique that you want to use, but there's sort of a lot of things that you need to decide on before you actually go and do it in the lab. So if anyone's got any ideas, that'd be really cool to discuss about. It's quite a sort of difficult question, I think, but I'm sure some of you will come up with some really cool ideas.
Yeah, we had so many great ideas for the first one, so it'll be great to see what we get now. Someone's jumped straight into the deep end on the questions with the ethics of it. So deciding whether your experiment is ethical, which I guess is going to be dictated by what it is you're studying. Someone said you'll have to decide what extra instructions you'd like to introduce into the template, so what that string of mutations or changes. Again, the size and type of mutations. Got a very good practical one of the cost of the experiment, which I like. It's not what I'd thought of, but yeah, if you've not got the money for it, you can't do it. Generally, what risks might be involved. So I guess that could range from the experiment not going right to any other risks. What type of cell you want to use, what metabolic pathway you might want to disrupt. Someone said cut out P53. I think that's they've got quite a distinct decision there. Someone's put what genotype or phenotype to investigate, what delivery method you want to use. So yeah, we've got some great experiments being formed in the chat box.
Definitely. No, that sounds, again, you sort of hit all of my ideas and more actually. So that's really great. Shall I, if the chat's sort of slowed down, I'll move on.
Yeah, if you want to go on to your next bit.
Great. So yeah, when designing a CRISPR experiment, I sort of came up with sort of 4 main questions we need to ask ourselves, but actually you guys came up with some really cool ideas as well that aren't included in this slide. So yeah, this definitely isn't an exhaustive list at all. But the first thing we want to decide on is actually what gene are we interested in editing. So for example, this might be a gene that we don't know the function of, or perhaps it's a gene which is commonly mutated in a disease and we want to work out why that is. We then need to decide what we actually want to do to this gene. So I think someone said this as well, but are there specific mutations that we want to investigate, or do we want to see what happens if we say change the expression of the gene or even remove it completely from the cell? So there's sort of loads of different things that we can do actually with our gene of interest as well. Next, we want to pick the model we use. So someone else said this too, but in the lab, we often use cells which are actually derived from cancers or stem cells, because these cells are often programmed to divide indefinitely, and so they're much easier to study. And so we might want to choose, for example, a specific cancer type if we're interested in how the mutation affects that cell type or that cancer type even. And finally, we want to decide what the readout for the experiment will be. So this means choosing what differences we're actually interested in seeing. So this experiment here is an example of a project that we might see in a lab using CRISPR. And looking at the title alone might be quite daunting. So it's genome-wide CRISPR knockout effects on lung cancer viability. But actually, if we break it down, we can sort of work out what this means and sort of work out what the experimental design might be. So to start with, genome-wide means that the experiment is looking at all genes present in the human genome. The next part says that the experiment will perform CRISPR knockout, and that means that each gene will be completely disrupted or deleted in the experiment as well. The experiment's going to be done in lung cancer, and it will also look at the effect of each gene and its knockout on how well the cell survives, so using cell death as the readout. So in other words, by using CRISPR to remove the function of each individual gene in lung cancer cells, we can look for genes which are important for lung cancer survival. And identifying genes which are important for lung cancer survival could be useful when designing new cancer treatments, for example. So let's walk through how this experiment might actually look in the lab. So we first start with our lung cancer derived cells, which we can grow in the lab. And we want to also design a set of guide RNAs which target every gene in the genome individually. And then what we want to do is we want to transfer these guides into our cells along with the Cas9 protein. So what we do is we aim to have one guide in each cell, meaning that only one gene will be knocked out in each cell. And this allows us to see the individual effects of genes being disrupted. Once we have our cells with the different edited genes, we can keep track of them to see if any of these knockouts have an effect on cell viability or how well a cell survives. And this can be done through what's known as a time course experiment. So what this means is that across time, as the cells begin to divide, this will increase the number of cells which carry each gene knockout. So when a cell divides, the DNA is copied, and so any edit that we've made will be copied into the daughter cell as well. So we would expect for each of these edits to sort of increase in number as the cells are sort of left over time and start dividing. However, if a gene knockout actually causes a cell to stop dividing or even to die, the amount of cells carrying this mutation will not increase. So we can compare the abundance of cells with each knockout after a set number of days and use this information to find genes which potentially affect cell viability. So let's compare here the cells at day 0 versus day 20. So I've got a question for you all again, but what can we say about the cells with the red knockout versus cells with the purple knockout? So again, if any of you have any ideas, pop them in the chat box and we can discuss it.
So we've got one answer just saying that the red cell died, which, yeah, we can't see any of those by day 20.
Yeah.
Just wait and see if we get any other answers. Someone's put it very nicely. So the red cell inhibited cell division and the purple one encouraged it by the looks of it. Yeah, someone's also put the mutation in the red cell caused it to be unviable, and the purple is thriving is my favorite summation of the purple one.
That's great. Yeah, that's perfect. Purple is more present. Great. So that's exactly what we can see here. So we can see that there's actually been a decrease in the number of cells with the red gene knocked out, but actually an increase in the number of cells with the purple knockout. And so we can use this information to sort of hypothesize that whichever gene we knocked out in the red cell has had a negative impact on viability or has caused it to die or not to divide. But actually the edit in the purple cell has had a positive impact on viability, and actually it's become the most populated cell type in this population. So this information is especially useful in cancer research, as what we want to do is we want to try and usually find things that cause cancer cells to die or limits their growth. So therefore, the gene that's been targeted in this cell could point to a potential cancer drug target. So it sort of shows how we can go from using CRISPR and sort of, we don't even have to target a gene in this case, we can just target all genes and say which genes are actually causing our cancer cells to misbehave or to die. And so we can go with an approach that we don't have any expectations. We're just going to test every gene and see what happens. Alternatively, we might say, actually, we're really interested in this gene, so we're going to do loads of different mutations and see what these different mutations do. And you can do a much deeper dive into sort of a single gene as well. So there's such a broad range that you can do with this technology, which is really cool. So we've seen how CRISPR can be used in the lab to further our research and understanding of gene function and also disease mechanisms. But gene editing also has the potential to actually treat diseases directly as well. And one way this has been done is in the treatment of a blood disorder known as sickle cell anemia. So patients with sickle cell have a mutation in the HBB gene, a single base change from T to A, and this is known as the HBS mutation. And this HBS mutation causes blood stem cells in these patients to produce red blood cells that are misshapen. And this can affect how these red blood cells transport oxygen around the body and also can lead to things like blood clots forming as well. Recently, however, there have been trials to use CRISPR to actually reverse this HBS mutation in blood stem cells captured from the sickle cell patients. So once the stem cells HBB gene has been changed from the HBS to the normal HBA sequence, these stem cells can then be actually transferred back to the patient, and normal red blood cells are then able to be produced. And this treatment has actually shown really good promise in its trials, but unfortunately it's still currently very expensive, and so isn't a widely available treatment just yet. There's also many other ways that people are trialing the use of CRISPR outside the lab. For example, this includes the modification of mosquitoes to no longer transmit the malaria parasite. Also editing crops to have a better yield or be drought or pesticide tolerant. This is similar to the golden rice example mentioned earlier, although I think that was using a different gene editing technology. And also even engineering bacteria to produce bioplastics and other useful materials as well. So it's got a really broad range of issues that it can be applied to. However, there's also many things which need to be considered when we're using gene editing in these kinds of applications. So, for example, if we use it to interfere with mosquito populations, this might have a negative impact on the wider population that we can't be sure of. So we sort of need to be really careful in general when we're sort of interfering with an ecosystem. And when it comes to genetically modified crops, there are some concerns that these might not be safe to eat, and many countries actually currently ban them. So whilst there's a lot of evidence to support the potential for gene editing and for CRISPR to tackle these issues, it probably will take a long time for these technologies to be approved and much longer for it to be approved for some uses than others. So with this in mind, let's do another quick poll. Do you think you would ever like to eat foods which have come from CRISPR edited crops?
That poll should be on your screen. And if you've got any reason that you've picked yes or no, you can also put those in the chat as well. And we've got a pretty overwhelming yes. So we're on about 70% yes,
Okay.
About 30% maybe, and only about 3% no. So yeah, looks like you've convinced people onto the onto CRISPR crops. Yeah, still overwhelmingly yes.
Interesting. Yeah, I think it's one of those things that it's like, I mean, and I think in my mind as well, I'm like, I'm sure I would, but then, you know, you're not really given that choice at the moment. So yeah, it's definitely a controversial question, and some people will definitely be no, and like for many like good reasons perhaps, but it's sort of, I guess, a bit of an unknown. Like whilst they've been shown to be safe to eat, some people might not like the idea of it still, which I think's, you know, fair enough.
Yes,
I guess,
someone's popped in the chat that you wouldn't know the long-term effects. I'm guessing that's why they may have picked maybe or no that
yeah, I think that in general is sort of a reason for a lot of people saying they wouldn't want to, because, and with other things as well, that even though we can say, okay, they're safe to eat, or, you know, scientists might say these are completely safe to eat, they've not tested the effects of eating them for 30 years because we haven't had that time to test it. So there's always those kinds of unknowns as well. So yeah. So I've just got one more sort of topic of CRISPR discussion before the Q&A. So another big topic you might have heard about is actually using CRISPR on editing human cells. So one question might be that if we know what mutations cause disease in humans, why don't we just use CRISPR to actually just get rid of them? And firstly, whilst we can edit cells in the lab, it would actually be impossible to edit every cell in an individual and actually eradicate all traces of a mutation. Instead, in order to make an edit which affects all cells in an individual, we need to make a change at a very early stage of development. And this means using CRISPR on human embryos. By doing this, all cells in the individual will be edited, but this also means that the gametes will be edited too. And this means that the genetic change could also be transmitted to future generations. So you're not just making the edit in one person, but also potentially in their children as well. And so this can be especially dangerous if we don't know what the effect the mutation will definitely have. And there might also be what we know, what we call off-target effects. And this occurs when other DNA sequences are altered during the CRISPR editing. And these changes might have, terrible consequences for the individual that are unknown at the time. So there's a lot of unknowns and risks that we don't mind taking in a cell in the dish, but actually taking them in a human is probably not the best idea right now. However, surprisingly, this actually has been done before. So in 2018, a Chinese scientist revealed that he had actually edited twin embryos using CRISPR. And the edit, in theory, it was designed to protect the individuals from infection of HIV, and was actually the first time that CRISPR-edited embryos have been continued into birth. However, the news of the CRISPR babies really was not well received across the globe, and the scientist was actually sentenced to 3 years in prison because of this, as it's currently illegal to do this. So, as we've discussed, whilst CRISPR technology is extremely exciting and it's commonly used in genetic research currently, there are many risks associated with it, which mean that it might not be suitable for use in humans just yet, or actually ever. And this is a very much an ongoing debate. And I'm sure that if you're interested, there is a lot of debate forums out there online that sort of argue the for and against as well. So that brings us on to the end of the CRISPR discussion. So if anyone has any questions, feel free to pop them in the Q&A now, and we can go through them.
Awesome. Thanks so much for that. We've had some really good questions that I'm going to pick out a few of. One that didn't even cross my mind, and I think it's a really great question. So you mentioned at the start that this sort of system was sort of copied or inspired by sort of natural immunity in bacteria. So someone's asked, could you write viral DNA from a known virus and insert it into a bacteria to give it natural immunity from that virus, even though it would never have encountered it naturally?
Yeah, okay. So that's something that is very interesting. And technically, yes, I suppose CRISPR could be used in that way, that we could engineer bacteria to say, so with the bacterial immune system, as this person's, I think, correctly inferred, when they encounter this of any viral DNA, they actually have a memory of it as well. And so they know that next time, that's a virus DNA, and they will cut it. So I think that, I'm not sure if that has been done, and what the uses of that could necessarily be, but I think that at least in my mind, I think that it would be possible to do that, definitely.
Yeah, it definitely sounds possible. It's one of those things that if you've not tried it, we don't fully know, but it sounds
yeah, I mean, there very well might have been people that have tried that out there, but yeah.
Someone's asked, what are the known limitations of CRISPR, which you sort of touched on at the end there.
Yeah, so sort of in recent years especially, as I sort of mentioned, CRISPR has only been around really for the past 10 years. And sort of at the beginning, there were a lot of problems with these off-target effects. So, for example, in the lab, because the DNA sequence in humans at least is so vast, there are often lots of regions which are quite similar to each other. And so you design these guide RNAs which will recognize a specific sequence of DNA, but it can be quite difficult to find a sequence which is actually unique as well. So we want to target this gene maybe, but we also don't want to target all these other genes which have that same sequence as well. So that's definitely one limitation. Another thing as well, which especially can be a problem in lab experiments, is that when the CRISPR actually cuts the double-stranded DNA, and the DNA repair mechanisms kick in, it's sort of like a stress response which kicks into the cells as well. And that can actually in itself change the behavior of a cell. So these cutting events can sort of stress out our cells and make them do things and maybe change their gene expression in a way that isn't actually related to the edit we're doing, but just because we're doing CRISPR at all. And so sometimes that can have an effect which sort of confounds our findings as well. So yeah, that's another sort of it's sort of known as like CRISPR toxicity, which is sort of a limitation currently too. But yeah, I think especially with uses of CRISPR outside the lab, these limitations are like even more important, because we really don't want to be making unknown edits in things we're going to eat or in human embryos especially. So yeah, we've got more room to have these errors in the lab for sure.
Someone's asked whether you talked about sort of editing human embryos. Could you use CRISPR on induced pluripotent cells as well, or instead of embryonic cells?
Yes, so similar to the sickle cell example that I spoke about as well, we can actually take stem cells out of the body and edit these cells and even put them back into the patient, and then they can go on to generate, in the case of the sickle cell, the blood stem cells will actually go on to create loads of red blood cells, and all of these will have the new edit in. So we can actually use CRISPR in the body, and we can use them on pretty much any cell type, including the induced pluripotent stem cells, which I think you mentioned. But yeah, it really can be. I mean, some cells are easier to edit than others, and this can be sometimes because of like how well they respond to the stress of being edited. But yeah, in theory, you can do a lot with it and do almost most cell types.
Awesome. We've had loads of questions come in, so I'm going to try and like sort of go through and
pick
out some particularly interesting ones. Someone asked sort of earlier, when you were talking about the two different when you're talking about sort of how CRISPR makes an insert, how you ensure that the template strand is used for homology directed repair as opposed to the sister chromatid.
Yeah, so this is a really good question as well, actually. So quite often what we'll do is on the template that we add in, we'll first of all add in a lot of templates. So we'll completely like try and saturate the cell with loads of this DNA template. So the idea is that when the cut's made and the cell comes to repair it, it will, the first thing it will find will be this template rather than the sister chromatid, like you correctly mentioned. But another thing we can do is actually in our DNA template, which has the new edit in, we can change the sequence so that once that edit has been added in, the guide RNA no longer will recognize it. So what will happen potentially if you cut the DNA and then it gets repaired by the sister chromatid back to exactly the same sequence that it once was, is that the guide RNA will still recognize that sequence, and so it might cause it to be cut again. But if we instead make sure that in our template DNA, we change the sequence enough so that the guide won't recognize it anymore, then every time the DNA gets cut and repaired, if it's repaired by the sister chromatid, it will just be cut again and cut again. But once it's repaired with the DNA template, then we can design that in a way that once that replacement has been done, it will no longer be cut, and so like that edit will be sort of set in place. So yeah, that's definitely a very sort of important problem that we've had to deal with in the lab as well. So really good question.
Sort of linked to the sort of designing an experiment side of things. Someone's asked how much would a typical CRISPR experiment cost to start to finish, and sort of how long it would be. So I guess that one that you described, that experiment of a knockout in cancer cells, any sort of ballpark on cost and time?
Yeah. So this has very much been a question that I've been asking my supervisor as well. And to be fair, what I've found is that the costs of research are, first of all, very expensive because there's sort of a lot of specialist molecules and chemicals that you need, but also are sort of, sometimes you don't even know how much something costs. You just know that you can order it or it can be done. So, but in general, so basically the most costly part of it, I'd say, would be actually ordering the set of guides you need. So basically, at least how I've done it before, is that you'll design all of these guides on the computer. So you'll have the sequence of all the guides you want, and then you'll send it off to a company which is able to manufacture all of these RNAs for you. And it will just send you back a little tube with like a tiny bit of liquid in, and all of your guides will be in that little bit of liquid. And that usually is the most expensive part of it. And they can that can range from, if you're doing only a very small, you only need a very small number of guides, then it will be quite cheap. It might only be like £100. I mean, I'm saying, when I say cheap in science, it doesn't mean cheap in real life. But it might be, £100. But if you want to do all the genes and you want to have tens of thousands of guides, then, that can go up to tens of thousands of pounds as well. And that's probably, in general, the most expensive part of CRISPR, I would say, is actually getting that order in. But it also depends on what your readout is. So depending on, if you're looking at, for example, just which cells die and which cells survive, that's quite a cheap experiment to do. But if you want to say, one experiment that I'm actually doing is, I want to look at how each cell, once I've edited it, I want to see how all of the genes in that cell are expressed. And so that's quite an expensive experiment to do. And so actually for me, that part's the more expensive part of it. But yeah, the bills can rack up, but it's all like, it's all useful. So, and I think with sequencing especially, like the prices have just come down massively in recent years. And so I'm sure because CRISPR is becoming, well, it's already such a massively widely used technique, the costs are, decreasing massively over the years as well, making it much more accessible.
And there's a sort of
Sorry, that was very long-winded.
No, it's a hard question. It's a hard question to answer, and it's not like you go and build a bespoke lab. You wouldn't go into our car park and start building a lab just for you. We work on a research campus. It already has labs and things set up, so it's difficult one to cost out from scratch. Someone asked to sort of follow up to that, saying, because it costs a lot, do you personally feel more pressured to get it right, or to get something to work the way you want it to?
Yeah, I think actually, at first, this is just on a personal level. I think it was quite daunting at first, being like, okay, this is a very important question that we're targeting. I mean, it's a different question to the one we spoke about, but actually in science, a massive part of it is learning that things don't go to plan at all, and that's actually completely fine, because you learn so much from if an experiment doesn't work the way you expect, that you almost always learn from that experiment in a different way anyway. And these costs as well, usually, for example, if you order in a library of guides, so all of your guides coming in a small tube, you don't actually just use that tube in one go. You can amplify that yourself in the lab for quite cheap. And so you've got a lot of room to sort of improve your experiment and sort of do trial and error and make sure that your experiment is optimized. So usually it's not like that much pressure in that sense. And I think most supervisors and everyone's very understanding that, you can't, these are unpredictable systems that we're working with. But so yeah, it's not, there's pressure in other ways, but I guess it's just how you see it. And I think especially in research, as I said, you just got to be prepared to learn from the things that don't go as expected, rather than seeing them as, oh, that's a failure, that's gone wrong. Like you can learn, oh, why has this not happened? Okay, that's something that we didn't know before. So it's always learning.
And I guess as well, you being a PhD student, before you even will have started your project, someone else has agreed that yes, it's a good idea that you do this project. So it's not like you just randomly chosen some expensive equipment to play around with.
Yeah.
It's been sort of, you will have done a lot of reading and understanding to make sure that you've tried to get rid of errors before you start. So I think that must help as well, knowing that it's not just you thinking that this is a good idea. Like collectively as a lab group, you'll have all been like, yeah, this is a good idea, regardless of what we learn from it.
Exactly. You're surrounded by, expertise in so many fields. There's always someone that you can like ask for advice who's done something similar before. So that's really helpful as well.
Awesome. I think for now we'll move on to your sort of little talk about your career. If we've got time at the end, we'll come to some more of these questions because there's been loads of really, really good ones, a lot around the ethics as well. But for now, I'll let you talk a little bit about how you got to where you are.
Sure. Okay. Thank you. So yeah, in these last few slides, I just will sort of share my journey into biology and how I actually ended up deciding to pursue a PhD in genetics specifically. So this slide just shows a timeline of my education so far. So I finished my A-levels in 2016 before I started a degree in natural sciences at the University of Cambridge. And in my third year was when I decided to actually specialise in genetics as my main subject. And this led me to apply to do a PhD at the Wellcome Sanger Institute. And I'm currently in my third year of there now, third year of 4 years total. So the first step I took into a career in biology was actually when I chose my A-level subjects. So I personally decided to choose biology, chemistry and maths because these were the three subjects I most enjoyed at GCSE. But often you don't actually need to do 3 science subjects to study a scientific degree. And many universities might only require 1 or 2 science related subjects. So this might be the same also for like scientific internships, college courses, or research jobs if that's like a better fit for people that don't really want to go to university as well. And there are plenty other, I'm sure you're familiar as well, but plenty other of science, more science related subjects such as physics, computing, psychology, all available at A-level as well. So my advice, I think, would be to just pick the subjects that you're interested in, because I think in my experience, I'm glad that I just went with the subjects I enjoyed most, and it makes, the studying a lot easier. But yeah, the next step was then obviously choosing the degree I wanted to study. So I decided I wanted to go to university. And so I was looking at all of the different degrees that are out there, which is quite overwhelming, especially in biology, because there's so many different biology related courses out there. So what I found quite helpful was actually to look at the different modules which would be covered on each course. And I think this information is usually available on a university's website. So I think it's quite good to pick a subject which has multiple modules which are interesting to you, because often there's an opportunity to specialize in one of these subjects later on and like further down the degree. So it's good to have like a lot of options that you're interested in, rather than just saying, I definitely want to do, I mean, if you definitely know what you want to do, then that's great. But I know I especially did not. But yeah, I think in my case, at least, like what I thought I wanted to do changed a lot during my degree. So it's good to have a course that has that flexibility in there already. So actually a big problem for me at A-level was I didn't know if I wanted to pursue biology or chemistry. And so this did lead me to consider a degree in biochemistry, but I still wasn't sure that that was going to be the right fit for me. So I felt like I needed more time to decide whether I wanted to be a biologist or a chemist in my career. So I actually came across a course that would let me study both. And this was the Natural Sciences course at Cambridge, which is basically a very flexible degree course that covers all major subjects, all major science subjects rather, taught at the university. So when studying natural sciences here, you get to choose multiple subjects to study simultaneously, and you don't actually narrow it down to a single course until you're in your third year. So in the first year, I chose to study 2 biology subjects alongside chemistry before deciding that chemistry was absolutely not for me anymore. So in my second year, I decided to do 3 biology related subjects. And this included biochemistry as well, which I was originally considering. But I decided that that wasn't for me and instead chose genetics, which I absolutely loved in my third year. So the genetics course in third year at the university I was at covers 5 main different courses. So this ranged from developmental genetics to plant genetics. And I just love the subject because it has like a great mix of cellular biology and biochemistry. And it just demonstrates how DNA has the power to shape diversity, evolution, disease. And it's just such like as we've hopefully seen today, like a very broad topic, which has lots of potential to be inserted in multiple issues we face. And so with that, there came like a lot of like many great discussions about like the social issues and political issues surrounding genetic research and technology as well. And I remember having a similar debate about, you know, the ethics of CRISPR as well in my third year course. So it was also my third year that I decided I wanted to actually continue research. And so I applied to the Sanger, which is a genetics research institute, as talked to you about, which is also in Cambridge and offers a 4-year PhD program. So I especially love the idea of being able to do 3 mini projects in the first year of the program before then doing the main project across the final 3 years. So basically I'm very indecisive and I like having a lot of choices. So both my undergrad and my PhD sort of were chosen because they have like a huge amount of flexibility, which was definitely beneficial to me, I think. But basically the Sanger Institute is structured across 5 different departments, all centered around genetics, but it includes human genetics, cellular genetics, cancer, pathogens, and a new diversity department as well called the Tree of Life Project. So there are like so many research questions and focuses to choose from. So I did mini projects in the human genetics and cancer department as well. And then I finally chose my final project to be in a cancer group, which I'm currently undertaking now. And I think I chose cancer because at university I became very interested in the idea of studying the genetics of cancer, because there's so many fundamental questions that can be explored with it. So for example, how mutations have completely different effects across different tissue types, and a mutation might cause a cancer to develop in one tissue type and not another, and sort of questions like that, which are, you know, really interesting. And there's so many different biological questions that like sort of come with that, and you can look at it from so many different angles on genetics, biochemistry, the signaling. It's just, yeah, I like the idea of being able to look at multiple lines of evidence to answer a biological question. So I now study in the experimental cancer genetics group, which is led by Dr. David Adams. And my project looks to define genes which regulate cancer behaviors, and this is using CRISPR and single cell gene expression analysis. So basically the project involves using CRISPR to disrupt specific genes in cancer cells, and then followed by the investigation of how each gene disruption affects the gene expression profile of the cancer cell. And we aim to identify genes which either change cancer behaviors in a way that can improve their treatability, or for example, reduce treatment resistance as well. So that's sort of the end of that part of the presentation as well. So if anyone's got any questions on that, or if we've got time to go back to the CRISPR questions as well, that's great.
We had a couple of questions about what A-levels you did, but then you answered that. So that was, those ones have been easily answered. Someone's a nice question. Is there anything that you wished you'd done differently in your educational path?
I think at the time, and when I was at university, I think the course I did offered a huge amount of flexibility, but it did mean that because I was doing multiple courses at the same time, it was very quite stressful to, like, you were switching from maths to chemistry to biology, which similar to A levels, you've got to completely like switch your brain to a different subject, and I found that quite difficult. So I think when I look back, I'm like, now I know that I really enjoyed genetics. I sort of think like, oh, maybe I should have just done genetics from the start and like not had to worry about doing all these multiple subjects. But when I think back to when I was at A level, actually I was leaning more towards chemistry. So if I hadn't have had that opportunity to try all the courses in the natural sciences course, I don't think I would have ended up where I am, which I'm really happy with. So it's sort of a mix of like, I wish I'd done it differently, but I know that I wouldn't have done. So I think if I could go back and speak to my younger self, I'd be like, do genetics, because that's really what you love. But yeah, it takes some time to figure it out, definitely.
Sort of similarly, someone said, which A level did you enjoy the most, and whether you can be whether just doing biology at A level is enough, or whether doing additional sciences you found useful.
Yeah, so actually at A level, I think I preferred I think I preferred chemistry the most. And I'm not particularly sure why that was now looking back, because actually at university I really didn't like chemistry much at all. But I think definitely you don't doing, just doing biology or doing one or two science subjects definitely doesn't hinder you at all. And it's important, if you are planning on going to university, and you maybe have some universities in mind, to maybe check their course requirements, just in case you are worried about that. If you specifically want to do, say, neurobiology, and some universities might require additional science subject, but some might not. So if you're considering doing biology or any science subject at university, for example, I would just have a check before you go to choose your A-level subjects. But if you're not sure whether you want to do science or history or English, so you want to do all three, or you want to do two, it's not like you're shutting off doors either, I don't think. So yeah, I would definitely, if you say you hate chemistry, I don't think you can definitely don't have to do it, or there's a lot of flexibility out there.
Having also done a biology degree, I'd agree that chemistry can be helpful, but yeah, if you absolutely hate it, then don't put yourself through that. Someone's said, sort of quite broadly, what advice would you give to someone who wants to do a similar career path to you? So if we've got many A-level students, is there anything you'd suggest that they look into or do at this point?
So I think what was quite useful for me was looking actually into what sort of a job after university might look like. So I think the PhD I'm in now is quite similar to hopefully research jobs I'll be doing in future as well. And it's very different from any sort of studying I've done before at uni or at school. And so when I was this was not quite in A level, but you could definitely look into this now if you were interested. But when I was in my second year at uni, I decided to do like an 8-week or a internship. So I know you can do them for maybe just like a week or two. I did one for 8 weeks, which was actually at the Sanger Institute. And that was sort of my first exposure to like what research was like in real life, and not just, biology in a textbook, but actually like how biologists actually work day to day. And I think that was really useful for me to sort of get an idea of, okay, this is sort of what a career like this looks like, and do I want to do that? And for me, it also helped being like, actually, I really like the Sanger as well, so that's why I applied there. But if you have any opportunity that also it's quite difficult, obviously, to go out and get an internship, or you might not have the time or ability to be able to travel there or anything. But I think there are like online there's some like online seminars about like a day in the life of a scientist and things, which I think would be quite useful, because I didn't really have much of an idea before I sort of jumped into it. And another thing is that, the typical like idea of a scientist in a wet lab, like in an experimental lab, that's just like one avenue that you can go down with biology and with like any science subject. And so there's actually like so many different options that if you're interested in the if you're interested in biology in general, but actually you don't like doing experiments, there's, you know, computational biology is like such a massive thing at the moment as well. And that's something that I jumped into with my PhD as well, which actually I've loved way more than I thought I would. So I think, yeah, being aware of the different sort of careers paths out there is quite useful for, you know, if you don't see yourself in one career, maybe you see it used, but you still love biology, like maybe you can see yourself doing other things. So it's useful to try and get that exposure now if you're able to.
I popped in the chat box just two things that came to mind as you were saying that. So there's the Nuffield research placements that are very competitive, and they do have quite strict criteria on who's eligible, but it would be worth checking. I think they're for year 12 students or sort of equivalent in other home nations. I think they are only available to UK students, but again, do have a check of that, and they do link you up with organisations to do work experience either virtually or in person, or sort of a mix. I've also added a link to the USTEM Awards, who's a partner that we work with, and so they're kind of like a Duke of Edinburgh award, but only in science. So you'd have to complete aspects in learning sciences, sharing sciences, and looking at sort of careers and stuff. So if you're not able to do stuff in person, which at the moment is still very tricky, that might be one way to at least show universities that you're really dedicated to sort of doing science, and gives you time to sort of share, to spend more time on sciences as well. One more sort of very A-level age specific question was whether you did an EPQ, or I guess knew anyone who did one.
Yeah, so at the time of doing my A-levels, but there was an option to do an EPQ, but I didn't actually do one in the end because for some reason, they weren't very popular back then. But I think that is a really good way of sort of showing that you're interested in a subject, like beyond just what you get taught at A level. So, I mean, obviously you don't have to do an EPQ on something related to a degree you're applying to, but I think if you want to like dive deeper into a specific research topic, maybe, not only is it like quite useful for you to like get an idea of what's going on in, you know, an active area of science, but it does show universities or wherever you're applying to that, you know, you are interested and have sort of gone beyond just what you need to do in your subjects at school. But I didn't personally do one.
But it might be that right now, if doing in-person work experience is harder and your school does allow you to do an EPQ, I can imagine that might be a really good option. If you can't get into a lab to do work experience right now, then an EPQ is probably a great alternative.
Exactly. I found that when I was writing my my cover letters for university, that I was sort of trying to find topics that I was like really interested in and reading about them, and then saying like, you know, trying to explain that I've done this extra sort of reading and research in my cover letter. But I think if you've done an EPQ, that sort of shows that you've actually gone beyond, and it's also when you come to write your cover letters and things, you've already got that there to say, okay, I already know about this subject, I can like use that as a great example, which I think is really useful.
Awesome. We are coming up to 5:45, so we're gonna wrap up there because we've got through probably about half the questions. We had so many great questions this evening. So thank you so much for joining us, Olivia. Your talk was awesome. I love learning stuff when we have speakers on. So yeah, this session was recorded, and so it'll be available on YouTube and our website either tomorrow or on Monday. Links that I've put in the chat box and links that will sort of generally help support learn this topic will also be in the email that you get from Eventbrite tomorrow. And if you have any sort of further questions, you can always ping us an email. I should have put my slides back up and I have seemingly lost them, but give me a second. So yes, you can always ping us an email if you think of anything else, and we can answer them. We're taking a break from Genomics Lite until the new year, because otherwise we'd be running one on Christmas Day, and I don't think any of us need that. So our next session will be in the new year. It will be looking at the human microbiome, so the interaction between bacterial cells and human cells in the gut. The link is on screen at the moment. But will also be in your follow-up email tomorrow as well. So thank you again, Olivia, and thank you everyone for coming along tonight and having so many great questions and ideas. I don't think we've ever had quite so many questions in a session. So thank you so much. And yeah, hopefully see some of you next time.
Thank you.